when it is giving the protection, so it is protecting, it is giving the flexibility, as well as at the same time it is protecting that particular tissue from any damage. Then next is absorption, secretion, and diffusion. So anything, if it want to absorb something from the outside, so that should be through a barrier. Okay, directly if it absorbs, so the tissue cannot tolerate the external environment. Okay, so from the surrounding environment, this epithelial tissue will protect and it will absorb the material from the outside. And if it is a gland, like as I have said, most of them will form glands. So glands usually, this endocrine glands, all this will secrete hormones. So even the secretions, and if anything is there from outside to inside or inside to outside, the movement is through the diffusion. So all these things are mediated by this epithelial tissue. Then filtration. So to filter the materials which are traveling from outside to inside and inside to outside. And also they will secrete a slippery surface which is called as mucus secretion. So mucus is very important even for trapping the pathogens. Okay, even in our nose, it will produce mucus whenever there is any allergen which is entering, not only allergen, if it is any bacteria, virus. So such kind of mucus layers are also seen in the internal organs. Okay, so these are the main characteristics and functions of the epithelial tissue. Then you have some special characteristics. So they are cellularity. That means these cells which are present in the epithelial tissue, they are very tightly bound to each other. Okay, you cannot see any intercellular space between them. If there is present also, it will be very minute. We cannot detect. So they are tightly bound to each other. And these tight junctions are by some proteins which will be expressive. Okay, they are the junctions which are involved in the attachment and communication between the cells. Then polarity. So polarity means like the detectable differentiation. That means if you say North Pole, South Pole, means you can easily distinguish these two. Means what you say here, you can call them like apical and basal surface. So that means it is nothing but the two distinct areas which you can easily recognize. That is nothing but the polarity. So positive, negative, like that. The clear distinction must be seen. So these epithelial tissues always have apical surface and basal surface. So basal surface is towards the inside and apical surface is towards the outside or at the top. And this basal surface is supported by the connective tissue. So both the epithelial tissue and connective tissue, they will form a basement membrane. I will show you the image. It will be easier. Then it is avascular. Avascular means there are no blood vessels. Okay, nutrients directly diffuse from the basal layer. And it is innervated. Innervated means it is supplied with the nerves. Okay, and regenerative. It is highly mitotic in nature. Mitotic means mitosis, cell division. So always it will be dividing. Therefore, it is regenerative in nature. So this is how it looks. So you can see the top one from here to here. This is epithelium and the bottom is connective tissue. Right? So here at the bottom you can see the nerve ending and capillaries in the connective tissue, which will be entering into the epithelium. And here the distinction between this epithelium and connective tissue, at the border you have the basal lamina and reticular fibers, and at the top, whatever the region which is there, that is basal region, and at the top, okay, So at the bottom end of the epithelium is basal region and the top end of the epithelium is apical region. And at the apical region, we have microvilli and ciliae, which is small hair-like structure. And this, each one is a single cell. Okay, I will show you. Sorry. So each cell, so whatever the nuclei which is present, so each cell is linked to the other cell by cell junctions or tight junctions, which will adhere. You have some proteins like desmosomes, like that. So you have the junctions where they will be tightly linked, right? So this is how it looks. So how the names of epithelia are given. So always the first name of the tissue indicates the number of layers present. So simple epithelium means only one layer is present, one layer of cells. You can see here, only 1, 2, 3, only single layer. Then stratified means more than one layer of cells. Here you can see in the figure. You have basal surface and the apical surface at the top. Then the second name or the last name describes the shape of the cells. So what are the different shapes? Squamous means the cells are like wider. They will look like a plate or scale. And the cuboidal means like a cube structure. And the columnar means it will be tall like columns. Okay, here you can see the images. So when you are giving the name, usually you'll have a first name and the second name. First name will tell you the number of layers and second name will tell you the shape of the cells. Like stratified cuboidal epithelium means stratified means multiple layers, cube-like structure, which is epithelium. Sometimes it can also tell you, you may not, the name may not be such simple. Okay, like first name or second name. Sometimes you may have some accessory structures like cilia, which will be present. So that also can be included in the name. Like goblet cells, goblet cells or cilia or keratin. So even this can be included in the name of the epithelium. Sometimes you can see like this, pseudo-stratified. Pseudo means false, stratified. So it is not completely stratified, but it is pseudo. It looks like it is stratified. And then transitional. Transitional means sometimes it looks like one type and other time it looks like another type. So it is in between these two. Okay. So even such names also can be seen. Okay, by looking at the name itself, you can be able to distinguish like what kind of epithelial structure it is. Okay. So first we will look into the simple squamous epithelium. Simple means a single layer. Squamous, it is a flat cells. So single layer of flat cells with disc-shaped nuclei. So where it is present? It is present in the endothelium, that is the inner covering, where you can see this in hollow organs. So what are hollow organs? They are visceral organs which are like a pouch, like our intestine, stomach, or it may be cavity like heart, bladder. So in such kind of organs you see this inner covering, and also we can see middle covering in the that is called as mesothelium. So where we can see this in peritoneal cavity, pleural cavity, and pericardial cavities. So what is peritoneal cavity? Which is the tissue seen in the abdominal wall. Okay. Then pleural cavity is between lungs and also underneath the chest. Okay, in between the lung and the, yeah.
Ma'am, can you repeat the
Okay, peritoneal. Yeah, peritoneum is nothing but a tissue which lines the abdominal wall. Within the abdominal wall, we can say this cavity, peritoneal cavity. Then pleural cavity is seen between lung and chest walls. Then pericardial cavity is the membrane which surrounds the heart. So in these cavities, we can see this mesothelium tissue. And it also covers visceral organs of those cavities. So visceral organs are nothing but which are important, vital organs, right? So in this heart, lung, abdomen, so all this we can see this type of epithelial tissue. So the function, what is the function of this simple squamous epithelium? It is for the passage of material by passive diffusion and filtration. So passive diffusion means no need of any energy or any gradient for the movement of materials. And it secretes lubricating substances from serous membranes. So serous membranes are nothing but those are the membranes that covers the body cavity which are not directly open to outside. Okay, that means only inside you can see. Are you getting? Serous membranes are the membranes which lines the body cavities. So the body cavities which are only present inside, which is not opened outside. Okay. So in that we can see this serous membranes. So they will be secreting some like mucous membranes will secrete mucus. So like that we will get some lubricating substances from these membranes. Okay, so these are done by the simple squamous epithelium. And they are present in the renal corpuscles of kidneys, alveoli of lungs, lining of heart, blood, lymphatic vessels, and ventral body cavity like serous membrane or serose. So in these areas, we see this simple squamous epithelium. So these are the structures, how it looks. Yeah. So this is a mesothelial lining. Okay, you can see. And this is simple squamous lining the walls of the capillaries, how it looks like. So these are the original pictures. The next is simple cuboidal epithelium. So simple means single layer. Then cuboidal means cube-like structure. So it's a single layer of cube-like structures with large spherical central nuclei. And it is mainly functioning for secretion and absorption. Okay, so they are present in the kidney tubules, then secretory portions of the glands, and also ovary surfaces. So these are the structures, how it looks like. You can see here, these are the simple cuboidal epithelial cells, the single ones. So this is how they look. The next, simple columnar epithelium. So like column shaped or rectangular shaped. So they are simple, means single layer. So single layer of column shaped or rectangular shaped cells with an oval shaped nuclei. So some will have the cilia at their apical surface and some may not. And some may contain goblet cells. Okay, so the main functions of the simple columnar epithelium are absorption and also secretion of mucus, enzymes, and other substances. And the ciliated, whenever the cilia are present, so they usually propel the mucus or reproductive cells by the ciliary action. So this is mainly involved in the secretion of mucus. Okay. Okay, so these are, they are present in the non-ciliated form. Okay, the non-ciliated form of this epithelium is present in the digestive tract, gallbladder, ducts of some glands. Whereas the ciliated form are seen in the lines of bronchi, then uterine tubes and uterus. So these are the images of how they are look. Okay. The next is pseudo-stratified columnar epithelium. So pseudo-stratified, sorry, just a second. So pseudo-stratified means, pseudo means false. Stratified means it looks like they are stratified or in the multiple layers, but they are not actually. Okay. So all cells originate from the basement membrane, but they will travel to the apical surface. So basement membrane is at the bottom and apical surface is at the top. So all cells, they originate at the basement membrane and they will form a tall cells which will go and reach the apical surface. So whenever we see, it looks like they are layer by layer, but they are not like that. They are almost single layer. Okay, they also contain goblet cells and they bear cilia. So what are goblet cells? I think there is a question. So they are from pluripotent stem cells. Okay, they appear like a cup-like shape. So that's why they are called as goblet cells. And their primary function is to secrete mucin. Okay, and they are involved in the mucus layer, right? So for the secretion of mucus. So these are the goblet cells. And they also contain cilia, okay, hair-like structure. And the nuclei lie at varying heights within the cells. So because I told you they arise from the basement membrane and they will go up to the top, that is apical surface, they will be very elongated. And the nuclei also may in some cells the nuclei might lie at the bottom and in some cells the nuclei may be at the top. So therefore it will give you the impression of stratification. That means whenever we look at a single instance, what we'll think is that they are maybe multi-layered, but actually they are single layered. That's why it is called as pseudo-stratified. Got it? And their function is mainly in the mucus secretion and propulsion of mucus by cilia. Okay. So the location of the pseudo-stratified columnar epithelium is, if they are non-ciliated type, they are present in the ducts of male reproductive tubes and ducts of large glands. Whereas the ciliated variety are seen in the trachea and upper respiratory tract. Okay. The next is stratified epithelium. So stratified means layers, multiple layers. So it usually contains 2 or more layers of cells. And they will regenerate from the below, that is basement membrane. And the major role is involved in the protection. So the layers, multiple layers, what they will be doing? They will be protecting the tissue. They will be protecting the organ. Therefore, this tissue is involved in the protection. And they are named according to the shape of the cells at the apical layer. So because they are multiple in layers, so from the top, whatever the top layer, that is apical layer which is present, excuse me. So based on the shape of that cells in that layer, so this name will be given. The shape may differ at the bottom layers. So first is stratified squamous epithelium. So many layers of cells which are squamous, means like a elongated, like plate-like structure are seen. And the deep layers of the cells appear cuboidal or columnar. So that's what I said. In inner layer, the cells which are present in the inner layer, their shape may be like a cube or column. But the cells which are present at the top layer, they are squamous in shape. Therefore, the name stratified squamous epithelium was given. And it is the thickest epithelial tissue which is adapted for the protection of the organ. So you have some specific types. So first is keratinized. Keratinized means which will secrete keratin protein. Okay. So in our hair and nails, we will have keratin protein, right? So this surface cells are dead and which will have the keratin, lots and lots amount of keratin protein. Whereas in non-keratinized cells, usually they are seen in the moist lining of the body openings, right? The functions of these stratified squamous epithelium are, it protects the underlying tissue in the areas subjected to aberration. So aberration is thus like scratching or damage, right? The next is the location where they are actually present. So keratinized epithelium are present in the epidermis of the skin, and non-keratinized, they form lining of esophagus, mouth and vagina. So this is how they look. So non-keratinized versus keratinized. Okay. Then transitional epithelium. Transitional means sometimes they will be in between the two types of epithelium. So basal cells are usually cuboidal or columnar, and superficial cells are like squamous or dome shaped in this transitional epithelium. They will stretch, okay, and they will permit distension. So in urinary bladder means it has to increase in size whenever the urine quantity is more, right? So therefore, for this to permit this distension, it has the ability to stretch. Yeah, any doubts? Okay. Then they are present in the lining of uterus, urinary bladder, and also a part of urethra. So you can see this is the top one is the relaxed state and it is a stretched state. So you can see when it is relaxed, the cells are bigger in size. So when it is stretched, so the cells are very small and the portion is increased. Okay. Then the surface features of this epithelium are, so the apical surface, it will have microvilli, which are like finger-like extensions of the plasma membrane. And they are abundant in epithelia of small intestine and kidney. Okay, because it has to, for small intestine, it has to absorb a lot, right? And even in the kidney. So it has to absorb, reabsorb the minerals. And they have to filter. So that's why they will have the finger-like projections for more surface area. Okay, so it will maximize the surface area across which small molecules enter or leave. They also act like a knobs, okay, to resist the aberration. Okay, so I told you aberration means like a damage, okay, scratching, which will be a scrapping, okay, you can call. So that will be prevented by this apical, this microvilli which are present in the apical surface. And also it contains cilia. So cilia is like a hair-like structure, as I told you. It is highly motile, means always moving. And it contains a core of 9 pairs of microtubules. At the center, you have a pair, single pair, which is surrounded by 9 pairs of microtubules. And the center portion, it is called as the axoneme. Okay, I will show you the image. Then each pair of microtubules are arranged in a doublet, means 2 are present. And the microtubules in the cilia, they are like centrioles. How the centrioles look like in the cytoplasm, in the same way these microtubules are arranged in the cilia. And this is mainly for the movement of cilia. Okay, it will form like a coordinated waves, like clockwise, anti-clockwise direction, they will be moving. So here you can see outer you have 9 doublets or 9 pairs of the microtubules, and at the center we have 2, okay, a pair of microtubules. They are again made up of dynein proteins. Okay, we have some proteins which are made up of, and this is mainly involved in the movement of this cilia. So here you can see the ciliary motion, how it moves. That is called power stroke and recovery stroke. How power stroke means how it is bending, and recovery means again getting back. And the mucus, how this cilia helps in the movement of the mucus. Okay. So again, if we go in depth, again we can study about the cilia, but just remember that cilia plays important role here. The next is connective tissue. So epithelial tissue is over. Next is connective tissue. So it is very diverse and it is abundant tissue. It has various classes: connective tissue proper, cartilage, bone tissue, and blood. So the components of connective tissue are, they have some cells. So which are again from tissue to tissue, they will differ. And we have matrix, that means fibers, which also differs from tissue to tissue. And the substances, ground substances like dermatin sulfate, hyaluronic acid, keratin sulfate, chondroitin sulfate, they will be differing from tissue to tissue. Okay. So they all have the common embryonic origin. That means they are all originated from the mesenchyma. So here we can see the classes of connective tissue. So from mesenchyma, we have got fibroblast, chondroblast, osteoblast, and hematopoietic stem cells. So from fibroblast, fibrocytes have originated. Then chondroblast, from chondroblast, chondrocyte. Sorry, chondroblast to chondrocyte. Then osteoblast to osteocyte. And hematopoietic stem cells give rise to blood cells and macrophages. Then from this fibroblast, connective tissue proper is developed. And from chondrocyte, cartilage. Osteocyte to osteosis, that is bone. Then from blood cells to blood. And from connective tissue, there are again some subclasses like loose connective tissue and dense connective tissue. So this loose connective tissue are seen in areolar, adipose tissue, and reticular tissue. Then dense connective tissue are regular, irregular, and elastic. Then the cartilage, it forms hyaline cartilage, fibrocartilage, and elastic cartilage. And from bone, we have compact bone and spongy bone. Then blood, it will form various types of blood cells. So each, if we like, it will become a very big topic if we study each and everything. But just to remember, from all these have a single type of origin, that is from mesenchyma. So first is areolar connective tissue. So it underlies epithelial tissue and it surrounds nerve and blood vessels. And it has the same type of structure and functions like other connective tissue. And in all tissues of the body, it will form a border. Okay. And the structures which are present within the areolar connective tissue, they help in support and binding of other tissues. They hold body fluids. So we have various body fluids in our body. So that will be held by this areolar connective tissue. Then it will defend the body against the infection, and also it will store the nutrients such as fat. So first is connective tissue proper. So it's a loose connective tissue which includes areolar, reticular, and adipose, and dense connective tissue which includes regular, irregular, and elastic. So first is areolar connective tissue. So they are like gel-like matrix. It has 3 fibers, collagen, reticular and elastic, which will support the tissue. And they will have the glycoproteins as the ground substance. Means that it is made up of glycoproteins and secreted by fibroblasts. And it includes fibroblasts, macrophage, then mast cells and white blood cells. So the main functions are, it will cushion the organs. It is surrounded, it forms a border to all the organs and it will give a cushion-like protection. And it holds and conveys fluids, the body fluids or tissue fluids. And also it has important role in inflammation, mainly protecting against the infections. And the cells like macrophages, plasma cells, mast cells, neutrophils, lymphocytes, eosinophils. So all these are the cells of the immune system which will be gathering at the site of infection site and they will be fighting against the infection. So all these cells are the like components of this areolar connective tissue. So they are widely distributed under the epithelium. They are in the packed organs and they also surround the capillaries. The next is adipose tissue. So usually they have adipocytes which will store the fat. They have nucleus pushed to one side of the cells by fat droplet function. Okay, the nucleus is at the one side, one pole of the cell. And it will provide reserve food fuel. So all over the extra fat which will be stored in these adipocytes. They insulate against the heat loss. Okay, and they support and protects the organs. So the location is under the skin, around kidneys and behind eyeballs and abdomen and breasts. Okay, where we can see more fat deposition. Then reticular connective tissue. So they are network of reticular fibers which are like loosely arranged and they are very soft. They have internal skeleton, soft internal skeleton and support other cells. So they are present in the lymphoid organs which are also again involved in the immune system. Like lymph nodes, bone marrow, spleen. Then dense regular connective tissue. So they are mainly primarily parallel collagen fibers and fibroblasts and some elastic fibers are also present and there is no blood vessels. They are poorly vascularized, means a little amount of blood vessels may be seen and they attaches muscle to bone and bone to bone and they will withstand great amount of stress and they are present in the tendons, ligaments. Here is a spelling mistake, you can say aponeurosis, SIS. Okay, so aponeurosis are nothing but they are tendon-like structures which are present in the muscles, around the muscles and fascia around the muscles. So it is also a connective tissue in collagen material which is surrounding the muscles. The next is cartilage. So cartilage is firm, flexible tissue which have no blood vessels or nerves. So it is a matrix which is made up of 80% of the water and it will have a single type of cells which are called chondrocytes. So these cartilages are 3 types: hyaline cartilage, elastic cartilage and fibrocartilage. So first is hyaline cartilage. So it is imperceptible collagen fibers. Sorry. So it is a hyaline made up of hyaline, which is a glassy kind of substance. And chondroblast, they produce this matrix. And in the chondroblast, they will give rise to chondrocytes. Okay, blasts are nothing but the cells which are not what you call procells means from which, like osteoblasts, from where osteocytes will be produced. Like here also chondroblasts, from chondroblasts, chondrocytes will be found. So these chondrocytes, they lie in lacunae. And the function of these hyaline cartilage is that supports and reinforces. Okay, it is giving the support to the organs. And then it will give like a, act like a cushion, so that it can resist the stress. So it is very strong and it can resist the stress. Okay. So they are present in the fetal skeleton and ends of the long bones, then in the ribs and nose, trachea and larynx. The next is elastic cartilage. So they are also similar like hyaline cartilage, and they have more elastic fibers. So wherever there is a need for stretching, okay, flexibility, these elastic fibers are present. So the main function of this elastic cartilage, it will give the shape, okay, and it will give the flexibility. So usually in the external ear and epiglottis, we can see this elastic cartilage. Then fibrocartilage. So it is also a matrix which is similar to the hyaline cartilage, and it has collagen fibers mostly. The function is it will give the tensile strength and it will absorb the shocks. So it gives the protection. And they are mainly present in the intervertebral discs and pubic symphysis, and also in the knee joints. The next is bone tissue. So it supports and protects the organs, and also wherever this attachment to the muscles are present, so there this bone tissue is present, and it stores calcium and minerals, fat, and the bone marrow is a site from where the blood cells formation takes place. Hemopoiesis will take place from this bone marrow. Okay. So mainly this bone tissue is present in the bones. So here from bone tissue, usually red and white blood cells will be formed. And they are mainly involved in respiration of transport of gases, nutrients and waste. Blood is mainly for transport of nutrients, and it carries oxygen and carbon dioxide, like for exchange of gases, and also it will carry the waste material. So this is mainly present in the within the blood vessels. So this is called as a fluid matrix. This is blood tissue. Okay, so it's a fluid matrix, and so it is not a particular connective tissue. It is like a fluid in nature. So it is not a normal connective tissue. It is atypical connective tissue. It is also developed from the mesenchyme. And it consists of cells surrounded by non-living matrix. Then muscle tissue. So we have skeletal muscles, cardiac muscles, and smooth muscles, and their respective tissues are present. So skeletal muscle tissue. So they are long cylindrical cells. It has many nuclei. So they are called multinucleate, and it has striations. Striation is it has like layer, you can see the layers. Okay, and the main function is voluntary movement and manipulation of environment and facial expression. And they are mainly present in the skeletal muscles, which are attached to the bones. Sometimes they are also attached to the skin. Then cardiac muscle tissue is mainly present in the heart. It contracts to propel the blood into the circulatory system. So they look like branching cells. They are uninucleate. Only one nucleus is present. It has striations and intercalated discs. They are like discs are present. Here you can see in the image. They are present in the walls of the heart. Then smooth muscle tissue. They are like spindle-shaped cells with a central nucleus. They have the nucleus at the center. They are arranged closely to form sheets and no striations. You cannot see any layers. And they propel substances along the internal passageways in their involuntary control. And they are present in the walls of the hollow organs. So I already discussed what are hollow organs. The nervous tissue. Nervous tissue, they usually transmit electric signals from sensory receptors to the effectors. That means from the neuron to neuron or neuron to muscle. That is called synapse. The action potential will be transmitted from neuron to neuron or neuron to muscles. So in the form of electric signals. So these are mainly present in the brain, spinal cord and nerves. And the main components are brain, spinal cord and nerves here. And there are two types of cells present in this tissue. Neurons, which are excitatory cells, which will get excited whenever there is a signal. And the supporting cells are neuroglial cells. So what happens when there is any injury to the tissue? So immediately inflammatory response will be generated. If any damage to this tissue, whenever the cells die, there will be an inflammatory response. So we have like, there are some characteristics, how they look. Collar, dollar, functionalizer. So that means rubor, collar, dollar and functionalizer. Rubor means redness. Then collar means heat. There will be a redness in the tissue. And then immediately heat will be generated. Then dollar means pain. Pain will be generated. And functionalism is loss of function. So these are the characteristics of the inflammation. So there is a redness or swelling. Then pain, heat generation and loss of function. So this is not specific. It is a local response. Anywhere this can happen. And this limits the damage to that particular site only. It won't spread. Okay. And immediately immune response. So it will take some time to develop the immune response. So suppose if the injury is by any microorganism, then the immune system will respond. The immune cells like neutrophils all will come to the site of infection and they will neutralize or kill the particular microorganism. So these tissues, how they will be acting throughout our life. So at the end of the second month of development, the primary tissue types will be formed whenever we are in the womb. Okay, and the major organs are, they form into their own places. Then they will start only growth. So first 3 months only, all the structure and organs, tissues, everything will be formed. From 4th month onwards, they will be just growing in their size and they will be maturing. Okay. In the adulthood, some tissues will regenerate. Okay. And some will form from the stem cells. With the increase in the age, that will reduce. Like epithelia will become thin and the collagen in the tissue will decrease. Then even the bones, muscles and nervous tissue, they will start damaging. Okay, damage and then nothing but there will be no growth. So shrinking will happen. And the poor nutrition and poor circulation, if it is present, along which is associated with the poor health. Okay. So this is about the general histology of like what kind of tissues are present and what are their characteristics, shape and like functions and structure. So now you might have got the idea like how the cells appear. Okay. Now based on this, the structure and function, we will select the cells for cell culture, tissue culture or organ culture. So next we will discuss about a basic introduction or history about the cell culture. So I will share that PPT. Okay, now history of animal cell culture media and reagent, culture of mammalian cells, tissues and organs. So briefly we will discuss today, and in the next classes again we will discuss in detail. So in 1907, Harrison, scientist, he first actually cultured the frog embryo nerve fiber in vitro. In vitro means within the lab. So he cultivated frog nerve cells in the lymph clot held by a hanging drop method and observed that the growth of nerve fiber in vitro. So that means what he has done is they used to include lymph clot or blood clot so that it will provide the nutrients. Now we are separately adding serum or amino acids, everything separately, we are included in the media. But at that point of time, they used directly the lymph clot or blood directly to culture the cells. So first he cultivated frog nerve cells and he cultured them to study the growth of nerve fibers. Then in 1912, Alexis Carrel, he took the explant of chick connective tissue and heart muscle contractile for 2 to 3 months. He also cultured. Then in 1916, Rose and Jones, he trypsinized and subcultured the explant. So what is trypsinization? Means we use the enzyme trypsin for subculturing. I will discuss each process in detail in the next classes. Just remember, like, what is the timeline of the development of the cell culture. Then in 1920s and 30s, Carrel and Ebling, they have subcultured the fibroblastic cell lines. From 1925 to 26, Strangeways and Fell, they have differentiated in vitro. That means they have cultured the organs. So for that, there should be a differentiation. Right? So that was actually performed by these scientists. So in 1940s, Kailova and other scientists, they have introduced the use of antibiotics in the tissue culture. So like penicillin and streptomycin. Now we call them penstrep. So penstrep includes both penicillin and streptomycin. Then in 1943, Ehrlich, he established L-cell mouse fibroblast cell lines, which is the first continuous cell lines. Then in 1949, Enders, they have grown virus in the cell culture. And in 1952, Delbecco, actually he was the one who has given the medium for the generation. So now we use the media which is called Delbecco's minimal essential media or Delbecco's modified media, DMEM. Okay, this was that the DMEM media was based on his name, Delbecco. He used trypsin for the generation of replicate subcultures and also he performed virus plaque assay. Then in 1952, Gay, he is a scientist who established the first human cell lines which are HeLa from a cervical carcinoma. So HeLa is from a woman who was diagnosed by this cervical carcinoma. So her name was Henrietta Lacks. So from that lady, these cells were isolated and they were subcultured. So till now, even we can use this HeLa cells for studying any like the action of drug or vaccine or anything on this particular cervical carcinoma. So based on her name, Henrietta Lacks, they have given the name HeLa. Okay. So in 1955, Eagle. So even this media, development of defined media. So even for this media, we have his name, EM Eagle's Minimal Essential Media. Okay. Then in 1959, Puck and Marcus, they have cloned HeLa cells on a homologous feeder layer. Feeder layer is nothing but, usually we will have flasks, culture flasks. So directly on the culture flask, we will have a matrix for which the cells will come and attach. But sometimes if the cells are not growing properly, we will add this feeder layer. Feeder layer means there are another type of cells. First, we will add that type of cells, and on that type of cells, we will add whatever the cells which we need to culture. So these cells will feed on that bottom layer and they will grow. Then in 1961, Sorial and Efuris, they have first done cell fusion by somatic cell hybridization. Then in 1962, McPherson and Stroker, they have established and transformed BHK21. Then in 1964, Klainsmith and Pierce, they have cultured pluripotency of embryonal stem cells. Then in 1965, Ham, even here also we have serum-free cloning of Chinese hamster cells. Then 1967, Huber and Cohen, they have used epidermal growth factor. In 1968, Stoker, they have given the anchorage independent cell proliferation. So these are all various protocols they have developed. Okay, in 1969, Metcalf, they have worked on the colony formation assay in hematopoietic cells. In 1975, Kohler and Minton, they are very important. Like they have generated monoclonal antibodies from hybridoma technique. Then in 1976, Ilmency and Mins, they have studied totipotency of embryonal stem cells. 1976, Hayashi and Sato, they have used growth factor, which is supplemented by serum-free medium. And in 1977, Nelson Rees and Flander-Meyer, they have confirmed HeLa cells cross-contamination for many cell lines. Then 1978, they have produced serum-free media. So 80 to 87, specialized cells were developed. Then 82, Darnell, they have studied the regulation of gene expression in the cells. Then 83, Evans, they have studied regulation of cell cycle. Then in 1984, Colin, they have produced recombinant tissue types, which is plasminogen activator in mammalian cells. And in 1989, Weinberg, they have studied all oncogenes, malignancy and transformation. 1990s, they have developed Kennedy and Cruz, Collins, they have developed laminar airflow cabinets. So in microbiology, we see this laminar airflows. But even in animal cell cultures, we have similar ones, but which are a little bit different. Then in 1991, Griffiths, okay, they have developed the Salk polio vaccine in monkey kidney cells. And 1998, Thompson, they have cultured human embryonic stem cells. And in 2000, Human Genome Project, which is very, very important. So Human Genome Project, it was aimed to sequence a complete genome of the human. So that was initiated, and which included many subjects like genomics, many techniques we can call genomics, proteomics, genetic deficiencies, expression errors, many were studied. Okay, so overall, this is the timeline. Okay, and still many more scientists who have developed various products, various protocols, which have discovered various techniques in the cell culture. So some of them were given here, and we can discuss each like aspect whenever we are discussing the respective topic. So if you have any doubts, you can just, if you don't understand, you can stop me and ask. Okay. So first thing is, like before culturing a cell. So what is culturing means? Simply we are growing the cells in the lab. Okay, usually the growth happens within the body. So that cells first we have to isolate and we are growing them inside the lab. So that is the cell culture. So for growth, see in our body, if their cells are growing, so how they are growing? They are taking up various nutrients from our blood. So we are eating food. So from food, whatever the nutrients, various metabolic pathways are happening inside. And the nutrients are excreted like amino acids, carbohydrates, all these things, they are supplied to the cells so that they will divide and they will grow. So whatever the things which are happening in our body or whatever the nutrients which are required, that we have to supplement to the cells outside the body, that is in the lab. So that is nothing but the culture medium. So the culture medium is the combination of ingredients which supports the cell growth. So what are the ingredients like nutrients, growth factors, hormones, and also these are like food for the cell. And we have to create the environment also. So what are the components of the environment? pH. The pH should be proper. Then osmolarity. If you see in our body, every aspect will maintain its own pH. Okay, we have a different pH for blood, different pH for urine, different pH in the stomach. So like that, the proper pH is maintained. So all those things we have to maintain outside, and osmolarity of the culture also. So here 3 basic classes of media are present, which are different based on their supplementation. So what are they? Basal media, reduced serum-free medium, and serum-free media. Sorry, reduced serum media and serum-free media. So it is based on the serum here. And which medium you have to use, that depends on the which type of cultures you are culturing. So that's why we have discussed all these tissues, cells, what type of tissues. Okay, all these things are to have a knowledge about what are the requirements for a culturing of particular cells. Okay. So what is basal media? Basal media is a defined medium that contains essential and non-essential amino acids, then vitamins, inorganic salts, organic compounds, trace elements, but they do not contain any growth supplements. So the examples are balanced salt solutions like phosphate buffered saline, PBS, which is very commonly used in the lab. Then DMEM, I told you, Dalbecose modified essential medium. And RPMI, 1640. So these two are, which are commercial media. Okay. So see, in the olden days, they don't know, like they don't have any commercial media. So that's why they have used blood clots or tissues directly for the growth of cells. But now commercial media are available. And here the term defined medium means we know what are the components which are present, which are defined. Therefore, that is called as defined medium. So this DMEM and RPMI also, they are available with or without glutamine. If you want glutamine, you can add that. If you don't want glutamine, you can remove that. Okay, like that, we have all these components in a basal medium, which is basic. So if you want anything, you can add them. Okay. Then reduced serum media means it is same basal media, but you have some nutrients and animal derived factors, and some amount of serum is also added. So we all know what is serum. It is the, whenever the blood clots, so the top portion is serum. So that serum has some proteins which are very nutritious, which are important for the growth of the cell. So small amount of serum can be added. And we also have some growth factors like epidermal growth factor, Okay, vascular endothelial growth factor. We have various growth factors. So based on our requirement, we can add. That is reduced serum media. Then serum free media. That means serum should not be added. Okay. It completely, the serum should be, because some cells you can add serum and for some cells you should not. And why you should not add also I will discuss. So serum-free media formulations exist for many primary cultures and cell lines, like Chinese hamster ovary cells, CHO, and hybridoma cell lines, Vero, okay, MDCK, MDBK, various cell lines. So why we need to add serum and why we should not add? If you add serum, so that is serum, it is very nutritious. It contains small amounts of micronutrients which we cannot add separately. Okay, it is almost a complete package of nutrients. So therefore, it will be, it has all growth factors. So it is very much needed for the growth of the cell lines. But why you should avoid serum? Because if you want to perform any experiment or if you want to treat the cells with any drug or vaccine or something, so we don't know what are the exact components of the serum. That is the main disadvantage. So are you getting my point? So the advantage of serum is it has all the nutrients, micronutrients, macronutrients, vitamins, minerals, all are present in the serum, growth factors, proteins, everything. So because of that, the cells grow very nicely. If you add serum, that is advantage. Disadvantage is, as you have many components, sometimes you don't know the small amount of quantities which are present in the serum. We cannot define each and every component. Okay, this amino acids are this much or carbohydrates are this much. Like that, you cannot distinguish each and everything. So therefore, if you are performing any experiment, that particular drug may interfere with the components in the serum and you may get wrong results. So that's why whenever we are performing some experiment, we should avoid adding serum. At that point of time, we use the serum-free media. Got it? So these are the common cell culture media which are used. One is minimum essential medium. Then GMEM, which is Glasgow Minimal Essential Medium, EMEM, Eagle's Minimal Essential Medium, DMEM, Dalbecos Modified Eagle Medium, then Medium 19199, then Basal Medium Eagle, Ham's F10 Medium, Ham's F12. This is all based on the names of the scientists. Okay, Ham's F12 Medium, RPMI 1640, then Lebowitz L15, CMRL 1066, then DMEM, Dalbecos Modified Eagles Medium 001, then MCDB and McCoy's. So these are the cell culture media used. And what are the components or reagents which are suitable for culture media? First is balanced salt solution. Then we need buffers and chemicals for maintenance of pH. Then cell dissociation reagents like trypsin, pepsin, so they will be used, and supplements like glutamine, because some supplements we will add. So these balanced salt solutions provide an environment to maintain the structure and physiological integrity, maintains pH and osmotic pressure. Also maintains osmolarity and regulate membrane potential because they'll have sodium, potassium, calcium, all this. And the ions are used for the cell attachment and enzyme cofactors are also present. So these are the balanced salt solutions. DPBS, Dalbecos phosphate buffer salines. HBSS, Hank's balanced salt solution. PBS, phosphate buffer salines. And EBSS, Earl's balanced salt solutions. Then the buffers used are Heppy's buffer and sodium bicarbonate solution. And the cell dissociation reagents, which will, whenever we want to subculture, we have to remove or detach the cells. So they are proteolytic or collagenolytic enzymes like peptide, trypsin. Okay, trypsin we have 1x and 10x concentration. Then dispase, then elastase, hyaluronidase, pepsin, collagenase, trypsin inhibitor. So all these are the cell dissociation reagents. Then other supplements like glutamine, L-glutamine, Glutamax, non-essential amino acids, growth factors, hormones, vitamins, mercaptoethanol, lipid supplements, CHO supplements, antibiotics, antimycotics. So all this will be used. Then serum. So serum is mainly from two sources: fetal calf serum or fetal bovine serum, which is for best growth conditions. And they are a source of growth and adhesion factors, hormones, lipids, minerals. And they regulate cell membrane permeability and serves as a carrier for lipids, enzymes, micronutrients, and trace elements. So how this culturing is done is, so we have cell cultures, tissue culture, and organ culture. So we will look into the tissue culture. It is used for in vitro cultivation of organs, tissues, and cells. So it is nothing but the ability to survive and grow tissue outside the body in an artificial environment. So this tissue culture is again of three categories. One is cell culture, explant culture, organ culture. Organ culture means the entire embryo or entire organ is removed from the body and it is cultured inside the environment, inside the lab. The organ culture is nothing but it is a three-dimensional culture. If it is cell culture means it is usually monolayer cultures which will be cultured in the lab. But here in the organ culture it is three-dimensional, maintaining all histological features of the tissue. And the tissue is cultured in the liquid gas interface. So it will be in a grid. So you have different flasks for cell culture, tissue culture, and organ culture. So usually you'll have like a bottles in which we will culture the organs. So advantage is, so whatever the physiological functions which are maintained in the body, same you can see in the organs. See, whenever you culture the cells and you get some result that may not function in a similar way in the organism, because cells are a single type of cells we will culture in cell culture. But in our body, you can't see a single type of cells. It will be usually combined with the other type of cells which are together functioning in a tissue or organ. So therefore, in organ culture, you can get the very exact results. Okay. And the cells, and also the cells, they remain fully differentiated. Okay. Because already they are differentiated in the organs, so they will remain differentiated. But the disadvantage is the particular cells of interest may be very small in number. Because even in tissue or organ, you can see many types of cells. So you cannot detect or you cannot study each and every type of the cell. So that is the disadvantage in the organ culture. So organ culture techniques. So you can see here the plates which are used. Plasma clot method means where in the plasma clot, that is hanging drop method, right? So or watch glass method, where the clot will be hanged and in that we will culture the cells or organ. And organ culture on agar gel method. So again, this is two types, raft method or grid method. And another one is in liquid media, complete liquid we will use. So these are the various techniques. The plasma clot method. So this was Dame, Honor, Fell and Robinson. They have used this plasma clot method, where plasma clot is acting as a substrate. Okay. And this is for the development of avian limb bone rudiment. For that they have used. So here you can see the image. At the bottom we have cotton wool. Above that we have a petri dish. And on that we have plasma clot. And on the plasma clot, the explant means the piece of tissue or organ that is cultured, that is placed here. Okay, so a plasma clot is prepared by mixing 5 drops of embryo extract with 15 drops of plasma in a watch glass and placed on a cotton wool pad. And this cotton wool pad is put on the petri dish. Okay, the time to time cotton is moistened so that it has to be so excessive evaporation should not occur. That's why we have to add some moisture to that. And then a small piece of organ tissue is placed on the top of the plasma clot to culture. So this is the plasma clot method or watch glass method. Second is the organ culture on agar. So here this agar is solidified and that is used as a substrate. So here we are taking 7 parts of 1% agar in basal salt solution and 3 parts of chick embryo extract and 3 parts of horse serum. 7:3:3 ratio we are taking. So here the embryonic cultures are grown well in this type of organ cultures. And here no need of any mechanical support as media here is the agar is giving the support and media is giving the nutrients. Okay, and we have another method which is called raft method. Here the tissue is placed on the raft of lens paper or rayon acetate which is floated on the serum in the watch glass. So here you can see in the watch glass you have a rayon acetate or you have a cloth or paper where you are adding the serum. Okay, and on that we are growing the tissue. And the rayon acetate rafts are made to float on the serum by treating their 4 corners with silicone. And in a combination of raft and clot techniques, the explants are first placed on a suitable raft and then kept on the plasma clot. So raft is nothing but it is for the support. Okay, so plasma clot is giving you the nutrients. Okay, so if you are adding the agar, so agar is giving only support, but the nutrient is given by the extract, embryo extract, right? And this modification makes the media change easy. Okay, then grid method. So initially it was devised by Trovel in 1954. So this grid method utilizes 25 millimeter by 25 millimeter pieces of mesh, okay, wire mesh, and or perforated stainless steel sheet also we can use. It is a grid. And the skeletal tissues are placed directly on the grid, but the softer tissues like skin, glands are first placed on the raft and then which are kept on the grid. Raft is for the support. And the grids are then placed in a culture chamber filled with the fluid medium. Okay, here like this. At the below you can see the image. You have the media on which the grid is placed. And the chamber is supplied with the oxygen and carbon dioxide for the growth. Then organ culture in the liquid medium. So this is about the support, that is solid medium. Now we can also perform the culturing in liquid medium. So the liquid medium consists of all the ingredients except agar. So there is no raft or no support here. When liquid medium are used for organ culture, generally a metal gauze or cellulose acetate or a raft of lens paper is used. So this is mainly for the nutrients are usually generated in the 3-dimensional way, okay, in the liquid culture. So what is the primary explant culture? So this was developed by Harrison and Carroll. So in the primary explant culture is nothing but, so a fragment of tissue, whatever the tissue extracted, that is directly placed on the glass or plastic liquid interface and will give the proper support or substrate, which can be a plasma clot. And then the attachment takes place and then the cell migration takes place. Cell migration is nothing but the growth. how the cells will be growing in the three-dimensional structure. So this is incubated in the growth medium. So here they can be on slide or cover slips, where single cover slip with plasma clot, or you can use double cover slips with plasma clot, or single cover slip with liquid media. And we have Carroll flask cultures. Usually the cell cultures will be done in this Carroll flask cultures. And we also have roller test tube cultures, which will be swinging. So in the slide or cover slip cultures, the slides or cover slips are prepared by placing the fragment of tissue on the cover slip, and which is inverted into the depression slide. I think you might have studied the mobility of bacteria by hanging drop technique. You will have the depression slide at the bottom, and above you have the cover slip. So same way here also we will do that. It's the oldest method of tissue culture, and still we can use them. So here a single cover slip with plasma clot can be used, where the medium is prepared in two parts, one containing 50% plasma in balanced salt solution, and the other is containing 50% embryo extract in serum. Okay, so here under aseptic conditions, we can use capillary pipettes and place 1 drop of plasma containing solution at the center of the cover slip. Like this, you can see. First place 1 drop of plasma, then add the tissue explant, and then add embryo extract, the another part of the medium, and spread the medium and place the explant. And then place 2 spots of petroleum jelly on slide so that it will seal. Okay, and we have to seal them, and we have to place them on the depression slide. Okay, where in the plasma clot it will get cultured. So transfer a fragment of tissue to this drop without crushing the tissue. Add the embryo extract solution and mix thoroughly before the clotting starts, and then locate the explant. Again, place the 2 spots of petroleum jelly near the concavity of the depression slide. Invert the slide over the cover slip and seal the cover slip. Allow the culture media to clot, and then turn over the slide and seal the margins with the paraffin, and we have to incubate that at 37 degrees centigrade. And double cover slip with plasma clot, where it same as the single cover slip method, where a small drop of balanced salt solution is placed on the cover slip, and then a square or round cover slip is placed on this balanced salt solution, and place the explant over it, and again another cover slip is added. The entire preparation is attached to a depression slide by petroleum jelly, and that should be cultured. Okay. Then single cover slip with liquid media, that is solid media. So in the liquid media, the explants are placed on the culture medium in a watch glass, and the explants are drawn into the tip of the capillary pipette, and one explant is deposited at the center of the cover slip. And the liquid medium is spread over this tissue as a film, thin film. And then a depression slide is added and is sealed with a petroleum jelly, and it is incubated at 37 degrees centigrade in an upright or inverted position. Then now, whatever recently we use flask cultures. So these are good technique for microscopic observation. So the advantages are the tissues can be maintained in the same flask for months or years. Okay, and large number of cultures can be done, and large amount of tissue also can be maintained here. Here you have 2 types of flask techniques. One is thick clot culture and thin clot culture. So thick clot cultures are suitable for short-term cultures, and thin clot cultures are used for long-term cultures. Here, first, place a drop of plasma on Carroll flask, and we have to spread them in the circle, and then transfer the explant and allow it to clot. And after plasma clots, the explants are fixed in a position and the extra medium is added. And these are incubated at 37 degrees centigrade with 5% carbon dioxide in the air. Then roller test tube cultures, where it will be always moving, okay? Movement is there. The feeding, patching and transfer of culture is done as in the primary explantation techniques, and this technique is used for preparing large number of cultures placed in the stationary racks or roller drums. The another name is roller drums. And here the cultures on plasma clots in the test tubes are prepared just like in the flasks. And in the plasma clots, these explants are cultured. So the cell cultures here, so these are the organ and tissue cultures. Okay. I will, if you have any doubts, I will answer at the end. So the cell cultures refers to the culture of the dissociated cells, okay? Or here, like they are separated enzymatically or mechanically, and they are made into suspension, then they are cultured, okay? But this tissue and organ cultures mostly they don't use. Cell cultures are more used in the normal labs, okay, for the development of cell lines. But after some generations, the characteristics will be lost in the cell culture. So that is the disadvantage. So in the next classes, we will look into the cell cultures mainly. Okay. So just remember what are the organ cultures and tissue cultures, what are the different flasks, what are the techniques which can be used. Okay. So in the next class, we will continue. So if you have any doubts, you can ask. What is, if you use roller, the medium will not disturb. So no, we are using plasma clots. So therefore, in the clot we are culturing. Therefore, the medium won't get disturbed. And regarding the practicals, it is not a single day experiment. So mostly complete practical may not come, but you may get the like by what you call that specimens, like you may get to explain what it is, but no need to perform all these things, because it is not a, maybe you can learn the techniques, but they are not the single day experiments for culturing all these things. Okay, but don't neglect. So you may get like a specimen, or you need to explain what it is. Yeah. Any more doubts? You can unmute and you can ask if you have any doubts. Okay, if there are no doubts, so I will be stopping the sharing. Okay. And we will continue the cell culture in the next class. Okay. Thank you so much.
Thank you, ma'am.
Yeah, thank you.